All right, so we're going to have a look at cell division today. The housemates are gone, so we're back on. So what we're having a look at specifically is the other purpose for why we would need to access DNA, and that's so that we can replicate all of the DNA so that we can use it for dividing. Division is a really, really key process for growth and repair. And sort of due to the complexities between both prokaryotic and eukaryotic cells, these processes, whilst similar in their essence or their outcome, actually have some key distinguishing differences. So the key processes that underpin them are a little bit different. And so we're going to have a look at, in this particular presentation, we're looking at mitosis and binary fission as two key processes, and then we'll get into meiosis a bit later on. And look at the key differences that underpin those. So binary fission is the process in which bacteria cells pretty much go from one to being two bacteria cells, so just basically that division in half. It's a pretty simple and straightforward process, and that's why it's different to eukaryotic cells or multicellular organisms. And essentially, DNA just replicates that circular loop of DNA that bacteria have, and everything else inside of it replicates, including plasmid DNA as well. The membrane starts to pinch together, and the chromosomes eventually separate apart to different ends, and then as the membrane pinches together, these cells will eventually pinch off and become two identical, daughter cells as such. So a very simple and straightforward process where there's a replication of DNA that occurs. The cell will elongate, so it becomes longer to allow for the additional material that's involved inside of there. A pinching occurs, so a cleavage furrow begins to form, as they call it, and then that continues to form until it is ready to pinch off. And so a pretty simple and straightforward way of replication, and the reason that it is simple and straightforward is because they don't have heaps of complex organelles or require heaps of complex structures. So yeah, I guess just understand that binary fission, key for bacteria. When we look at mitosis, though, we're now referring to cell division and the replication of DNA for the purpose of cellular division in eukaryotic cells, so multicellular organisms. This needs to be a distinctly different process because we have a lot of complex organelles inside of our cells, and there's a lot more DNA that is found, and the DNA is also organized differently as well, and so we need to be able to have different mechanisms for replication and for splitting apart, and things like spindle fibers and stuff like that to make sure information goes where it's supposed to go. And so mitosis, really, there's these certain phases that occur, but one of the key ones that occurs right at the very start is the interphase process that occurs before mitosis. And so interphase itself is kind of important because that's where we replicate DNA. It's where at the start point, or the gap one, what happens is any sort of key materials that are going to be required, such as certain proteins or key organelles that are required for the new cells, are replicated first. So all of that stuff is done. The G0 on that picture refers to maybe that cell was told, it's a mechanism where that cell was not told to replicate. So we say that that's cell cycle arrest. That cell was instructed by a particular protein reaction that do not replicate at this point, so it doesn't go through interphase. If interphase does occur, though, in the gap 1 phase, that's where we're replicating or creating key proteins that we need, setting that sort of those structures up, so like getting chromosomes ready for division and things like that, and getting all the other cellular contents ready, so things like mitochondria and stuff like that ready to go, and replicated more and more, as well as making sure that things like all the necessary nutrients are in place. In the S phase of the interphase of mitosis, that's where the replication of the chromosomes actually occurs. And then once that is done, we've got twice as much DNA inside of the cell at that point in time. And then in the G2 phase, that's the last phase of interphase, where it's almost like just a double checking, like are we good to go? Are we good to we've got the okay to go through with mitosis now, and then mitosis is the splitting.